Well, good evening, everyone, and a very warm welcome to the third of our COVID Connections discussion events, which are brought to you by Wellcome Connecting Science and the COVID-19 Genomics UK Consortium, or COG UK. My name is Ken Skeldon. I'm the head of the public engagement team at the Wellcome Genome Campus. And just to say that we've also got closed captions available for this evening's event. And so to turn those on, you just click on the live transcript button, which should be next to the Q&A box on the Zoom screen, and then choose Show Subtitle. So hopefully that will work for those of you who want to take advantage of that. Just on the series itself, so throughout this series, we've been exploring the coronavirus pandemic through the lens of genomics and understanding the virus at the level of its genetic makeup. And how that's been so vital in how we keep one step ahead of it. And so all our speakers have connections to the UK-wide genomics effort in one way or another. And that comprises a mix of institutions, universities, public labs, NHS centres, research campuses, including the big effort that's going on at the Wellcome Sanger Institute, which is part of the Wellcome Genome Campus, where we're hosting, at least virtually, tonight's event from. Also, as with previous events, tonight's very much a conversation. It's not just between our panel, but also more importantly with you. So there'll be lots of time for questions and reflections. And so please use the Q&A feature, not the chat feature, but the Q&A feature on the Zoom page, which you'll either see at the top or the bottom of your screen, depending on the device that you're using. And don't worry if it looks like, we always say this, but don't worry if it looks like you're the only person feeding in a question, because what's happening is they're coming into a feed that we have a look at, and because we get lots of questions, we try and cluster them and send those onto the panel. So please don't wait to the end, just do that as we go, and we'll try and get through as many of the questions as we can. So, so far tonight, we're delighted to be joined by well over 150 people and more joining as we go. And so just to briefly first welcome our panel. Just briefly, because our chair for tonight's event will give a proper introduction in a few moments. But tonight we're delighted to be joined by Kirsten Mayer from the Wellcome Sanger Institute, Michael Morgan from the Cancer Research UK Cambridge Institute, which is at the University of Cambridge, and also Kenneth Bailey, who's from the Rosalind Institute, which is part of the University of Edinburgh. And onto tonight's chair, and I'm really delighted to welcome Angela Saini. Angela is an award-winning science journalist and broadcaster. She's presented science programs on the BBC, and her writing has appeared in The New Scientist, The Sunday Times, National Geographic, and Wired. As the founder of the challenging pseudoscience group at the Royal Institution, Angela still is an active researcher and campaigner around issues of misinformation and disinformation, which could hardly be more relevant for the COVID-19 pandemic. And her latest book, Superior, The Return of Race Science, was a finalist for the LA Times Book Prize and named Book of the Year by The Telegraph, Nature and the Financial Times. Her previous book, which was called Inferior, How Science Got Women Wrong, has been translated into 14 languages. Angela has a master's in engineering from the University of Oxford and was a fellow at the Massachusetts Institute of Technology. And in 2020, she was named one of the world's top 50 thinkers by Prospect magazine. So I'm really looking forward to tonight's event. I hope all of you are as well. And without further delay, I'm delighted now to hand over to Angela and the panel for tonight's event, which is called From Mild to Mortal, Unlocking the Mysteries of COVID-19 Symptoms.
Thanks so much, Kenneth, and welcome to all of you at home. Thank you for joining us for this special public event. So this evening, as Kenneth said, we're looking at the mysterious nature of COVID-19 symptoms and how scientists are trying to understand them. What we know is that there are so many different ways in which the virus can affect and infect an individual person's health. One might become seriously ill, another may show no symptoms at all. How much of this has to do with our genes, we don't know, but today we'll be exploring whether and how genomics can help us to understand some of those differences in susceptibility to the virus. We have an hour and a half in total, or just less than that, and that includes questions from you. So the more questions that come in, the more time I'll leave aside for Q&A at the end. So please ask whatever you like. There are no questions too silly or too simple. This is really complex stuff, as you can imagine. And in some ways, because it's new science, it makes understanding it makes children of us all. So we're all learning together. Use the Q&A box at the bottom, not the chat box, and I'll read them out as they come in. So we have three wonderful leading experts to give us their thoughts, and I hope we can see them on the screen now if they'd like to put their videos and mics on. We have Dr. Kerstin Mayer, who joined the Wellcome Sanger Institute in 2017 and is now Principal Staff Scientist in the Cellular Genetics Group. She leads the Human Cell Atlas projects, which characterize the cellular landscape of the lung and the respiratory system in human development and over the lifespan. So that's a perfect specialism to be addressing this question. And she has studied diseases such as asthma and, of course, also COVID-19. Dr. Kenneth Bailey leads a research program in translational applications of genomics in critical care at the University of Edinburgh's Roslin Institute. He works as a consultant in the intensive care unit at the Royal Infirmary, Edinburgh, and during the COVID outbreak, he's contributed to the design and delivery of the recovery trial, which is looking for treatments for adults. So his research has helped lead to new biological mechanisms underlying critical illness in COVID and help contribute to the discovery of effective drugs. Dr. Michael Morgan has a PhD in genetics and is a computational biologist at the University of Cambridge. His research uses computational genomics, so he works with mathematicians and physicists to understand how cells respond to changes in their environment with a focus on the human immune system. And Michael, if I could just come to you first then. There are two sides to studying a virus. There's the genome of the virus itself, and then there's the genome of the host. Can you explain this difference and what you focus on in particularly in that equation?
Of course, and it's an extremely important distinction to make, and I'm sure that the panelists here would 100% agree with this, that a lot of the efforts that are kind of in the public domain and that are sort of in the news a lot are about the genetics and the genomics of the virus itself and how that's evolving and has evolved over time, and particularly over the course of the pandemic. It's been raging for 18 months. Now that's one side of things, is how the virus behaves. The other is how our bodies behave and react to that infection. And so the work that we've been doing is using genomics to understand what happens in our bodies, basically. So the cells that make up our immune system, that make up our organ systems, and Kerstin has been part of this effort in terms of the lung-based work, is how do those cells react to that viral infection, and what does it do to our physiology?
One of the interesting things here is that people respond so differently. I mean, even we know that everyone's genome is different, but for this, with this virus in particular, we've seen such different symptoms. How is that possible?
I mean, that's it's the kind of the question that's I guess is on everyone's lips, really, and the answer is very complex, and to be brutally honest, we don't know all of the answers. We're sort of scratching the surface using genomics at the moment, by looking at the way the individual types of cells in the immune system react to the virus and how they communicate with each other, and how that those complex interactions shape the response to the viral infection. Now, the immune system, and this sort of always comes up with immunology, the immune system is incredibly complex. And that's in part because there are many moving parts, and those moving parts often react in different ways depending on the environment. So it sort of gives you an idea of the complexity involved. But there are some things that we have sort of got a better understanding of. For instance, the types of cells, types of white blood cells that you'd expect to see during the course of a normal infection. Well, what we can do is we can compare those things, those cells, and the sort of the rise and fall of those cells over time in a normal infection, compare it to what happens when someone's infected with the coronavirus, or this particular coronavirus. And that's some of the work that we've done, and Kirsten was also involved in this, in collaboration with groups in Newcastle and in London as well, and really a sort of a very large consortium, multidisciplinary consortium, where we found that particular types of cells that are involved in making the antibodies, but also the cells that help them make the antibodies. We saw that in asymptomatic patients, those particular types of white blood cells were elevated. And that suggests that those individuals are mounting a proper immune response to the virus, and they're able to fight it off very quickly. In contrast, the patients that were really poorly, the ones that were in ITU, the ones that Kenny would see sort of on a day-to-day basis, those patients, we saw a really delayed reaction in those particular types of white blood cells, which maybe indicates that prior to the infection, there was some variability, some differences between individuals that almost predisposes them to that differential response to the viral infection itself.
Then Kenneth, just coming to you then, how is there any way, because we think of genomics as, this is something that you can know something about a person to begin with. Can that then predict what their response to a virus will be, or is that just impossible?
Well, and as Michael said, it's early days for explaining individual susceptibility, but with the evidence we've got, I think we know that, as for other infectious diseases, susceptibility to COVID is in part encoded in your genes, but that only accounts for a relatively small amount of individual risk. And by far the biggest effect on an individual's chance of becoming critically ill or dying from COVID is age. So the difference between someone under 50 and someone over 80 in risk is 11-fold. And for the strongest genetic effect that we've discovered in a large UK study called Genomic, the effect was about 2.3. If you carried that genetic variant, you're about 2.3-fold more at risk of ending up in the intensive care unit. And it's a relatively unusual gene, so that doesn't explain much of the variation across the whole population. So I think the role of host genetics, studying human DNA to try to understand this virus, right now, the much more urgent application of that, rather than explaining a fascinating mystery, is to cut through the complexity of the immune system that Michael described. I'm not an immunologist, but I think Michael and Kathleen will agree that the immune system is probably the most complicated system in the body. It's probably become that way because of the constantly changing range of pathogens that we have to fight, and because those pathogens themselves interfere in the function of our immune system. And we don't understand it, despite hundreds of years of study. We don't really understand the mechanistic function of the immune system, certainly not in this new disease. And what genetics can do is cut through all of that complexity and take us straight to the components of the immune system where a change in that component, in that gene, changes the outcome for the whole patient. And that's potentially very important therapeutic evidence. It can tell us what targets we need to be aiming for with drugs.
So if genetics or genomics can only account for some of the differences that we see between individuals, then what are the other big factors aside from age that you see make a big difference?
So the big ones are age, number of other illnesses that you have, sex, so men are greater risk. And then there are probably, and I'll hand this question over to the experts on the line, but prior exposure to other viruses, I think probably has a big impact, and other factors that shape your immune system that aren't inherited from your parents. But actually, I'd be very interested to hear what the others in the panel think about that last one.
Okay, well, let's go to Kirsten then. And I just so the audience knows, Kirsten is in an area at the moment where her internet connection isn't very good. So she's dialing in through the phone. So even though you can't see her, we should be able to hear you. Kirsten, are you there?
Yes, I'm there.
Yeah, you are.
So,
okay, great. Wonderful. I'm glad. So to your mind, how can genomics help answer this question of these mysterious difference in symptoms that we see between individuals?
Well, I think genomics has become a fantastic tool to study the immune system in more detail. In particular, I've been working on something called the Human Cell Atlas, and that's allowed us to really drill into the different cell types in the body, and really not just look at blood and say, well, the blood responds as a whole, but really dig into the different types of cells within the blood and see how they all react in different ways, and subcluster cells into responders and non-responders in a way that we really haven't been able to before. And that's true for the immune system in the blood, but it's also true for the airways and the lungs, where we're getting a better understanding of what the cells are that have a role. And then also we're able to really see which cells can sustain replication of the virus. So, you know, which cells express the receptors that the virus can latch onto, so it can get into the cells and then cause the replication and amplification of the virus.
Sorry, how precise is that? How useful can it be when it comes to diagnosis or treatment, for instance?
So, for example, we can now really understand the different cell types that the cells that are activated, and I won't go through all the different cell types that might not be so useful, but the question came up earlier, you know, why are different people so different? And, you know, have maybe some people had exposure of a particular virus that might influence it. So one of the things we've been studying in our group is the difference in immune response between children and adults. Now, the immune system in children is very different from that of adults. It's called naive. Children are born and they haven't had any infections at all. So a lot of their immune system, especially what's called the adaptive immune system, that is able to mount a very specific response to the shape of a virus, is still very uneducated, if you like. It's very, very diverse to give the child the best chance of recognizing all sorts of pathogens that come along, but none of these cells have been expanded particularly because, of course, the child hasn't seen these viruses. In contrast to that, adults will have had a lifetime of exposure. So that means certain cells are expanded enormously. And then when a very common cold virus comes along, that adult is incredibly good at you know, reacting to a particular cold and very quickly neutralize that infection. But in the case of COVID, this is a bit of a double-edged sword, because some of these expanded cells can now react to the virus without necessarily actually neutralizing it properly. And you can then have an activation of a lot of these expanded clones that then becomes pathogenic. The cells expand, are activated, and cause some of the cytokine storm some of you heard about. But in children, we found that because there are no such expanded clones, we don't actually get that kind of cytokine storm and expansion, but we see only very little expansion of cells that are probably quite specific to the virus that will presumably help to clear the virus very quickly.
So does this then explain why children tend to be asymptomatic, and whereas the older you get, the more likely you are to show symptoms?
There actually is a second part to that story, and that is our immune system has really two sides to it. This adaptive immune system that I've already talked to it about, but also an innate system. And that innate system is really a way of responding to danger. And one of the things we found in our study, that actually in the airways of children, this innate response to danger signals, basically signals that are common on viruses and bacteria, is much stronger in the airways in kids than in adults. And the response of those cells is usually to produce antivirals, for example, interferon. And this interferon response is stronger in kids it stops the virus from replicating, and that means they can then clear the virus more easily.
That's okay. Because it does feel counterintuitive. It does often feel as though kids pick up bugs more quickly, but then you're saying in the case of this particular virus, that's not the case. It actually works the other way around.
That's right. So
yeah.
We don't really fully understand that yet. But one of the ideas, one of the thoughts is that this particular virus has a way of suppressing the interferon response. We don't quite understand how that happens, but it appears that in children that suppression is less effective, and therefore kids can clear it better.
Thank you. Mike, then just coming to you, I think a lot of people imagine, you know, when we talk about genomics, this to be a kind of fixed quantity that you are born with your DNA and it doesn't change. But in fact, it's much more complicated, the way that genomes interact with the environment and can change the way that individuals respond depending on their immune systems. Can you explain that?
I will certainly try to. Kenny touched on this a little bit when he was talking about what the differences in risk factors were with respect to COVID severity, and the fact that age is far more dominant effect than a single genetic variant is. So the complexity comes down to the fact that although we're born with a relatively fixed genome, modulo what we call somatic mutations, those are the mutations that occur in our cells over time as we get older, and they're sort of the things that we think about in terms of cancer risk and that sort of thing. We sort of brush those under the carpet a little bit in this context when it comes to infections. Things are complicated enough as it is. But the point really is that a genetic variant might be present, but it might not manifest the effect of it necessarily until your body experiences a very specific environment. So for instance, someone might have a particular genetic variant that predisposes them to weight gain. Now, if they live in a particular part of the world where they don't have access to very sugary or very fatty foods, then that individual isn't going to necessarily have a higher weight than someone without that particular genetic variant. So it's the environment and the genetics that come together really to create what we call the phenotype. So that's the manifestation, that's what we see or what we measure as scientists. A phenotype could be height, or it could be hair color, for instance. Or in this case, in COVID, the phenotype is how do you respond to the virus, and whether you clear the virus and are asymptomatic, like the children that Kirsten described, or they get very poorly and end up in ITU. So although we might be born with these sort of fixed quantities, so to speak, it's not necessarily deterministic. And I think that's a really important point to kind of drive home, is that you're born with something, but there are still ways to deal with it in essence, to modify things. It might be that a change in diet or a change in behavior can really drastically alter your risk or for a particular disease, and for COVID. Exercise, I'm sure, is almost always good for everything. So maybe that's one way to modify maybe your genetic risk. The easiest way to modify your risk is not to be exposed, obviously, to the virus. But even if you do get infected, there are other things that can be done to kind of mitigate the genetic lottery, so to speak.
It's been an interesting year in terms of science journalism, because I have seen lots of stories that have implied that there are certain genes that make some people more susceptible or make them more likely to have a worse outcome than others. So, for example, there was a story in the New York Times, I remember last year, around this Neanderthal gene. So there's a certain set of gene variants that people have that are linked to Neanderthal ancestry back in the midst of time, and they were saying those people have a different kind of susceptibility. How do you feel about those kind of claims?
I mean, the veracity of any claim is very much going to be contingent on the weight of the evidence. That's what we do as scientists, and it's why it's very important for scientists to get involved in engagement events like this that the Wellcome have put on, in order to be able to get across the importance of being able to critically evaluate those stories and that evidence. Now, I don't know about that particular story, about how strong the evidence was for or against necessarily, in terms of whether those particular variants. I think a similar story has come up that involving a similar class of genes, about why men might be more susceptible to severe COVID, for instance, because the gene is on the X chromosome. So women have, or biologically, women have two X chromosomes, and men are biologically born with just the one. And so it means that if you've got a particular variant, and you're a man, that means that there's no compensation on another chromosome, basically. And I think the sort of the stories are the same. It's sometimes it's very easy to make a just so story, but you base your conclusions on the evidence that's in front of you. And what's really important as well is that when new evidence comes to light, you're able to adjust your beliefs and your conclusions, when evidence might contradict or override some sort of previous conclusion. So it's a slightly woolly answer, and it's probably a very typical academic answer in that sense.
No, that's fine. So Kenneth, just coming to you, because you did say earlier that you see more men coming in than women who are critically ill, that their risk is higher. What do you ascribe that to? Why is that?
I don't know. So the evidence for that is quite strong. The UK has done some of the best clinical research in this field. There's a study that I co-lead called ISRIC-4C, also led by Callum Semple and Peter Openshaw and Liverpool College, respectively. And one of the early papers that we produced carefully described the risk associated with age and sex and other illnesses. The paper was written by one of my colleagues, Anne-Marie Doherty. The mechanisms of that, I think we can only speculate about. The specific genes that Michael mentioned that are on the X chromosome are potentially part of the explanation. The effects of hormones like testosterone and estrogen on the expression of proteins on the surface of cells are a possible mechanism. But teasing these things apart is difficult. What's particularly difficult is going from an association where you can see these two things go together in a population, being male and getting sick with COVID, for example, to saying this one thing causes the other thing. And that's a very difficult question to answer at a population level in humans. Our ability to intervene, to change things in the humans is restricted essentially to treatments that might help them, because obviously we're not going to go around experimenting on sick people just to better understand the disease. In order to answer those sorts of questions, scientists use randomization. That's the trick that was discovered years ago. Really, it's kind of part of the driving force behind the Enlightenment, and all of the progress we've made since then is an understanding that the natural world is always more complicated than we think it is. And randomization enables us to match up all of the things we know about between two groups of people or animals or whatever, and all the things we don't know about, all the things that matter, that vary in those populations that we haven't measured or don't even know exist. Where we do a drug trial, we randomly assign people to one drug or another, and we can see which group of people do better. And then we're really on solid rock. We know that the better course of action is to use or not use the drug. And that's really why we've made so much progress with treatment in COVID. I bring that up because we can use a genetic trick called Mendelian randomization to answer some of these questions. And it's sort of a fudge because we haven't answered the male question, but there are a couple of aspects of human biology that have been associated with a causal role in making your COVID worse using this method, and they are smoking and obesity. A really big international consortium called the Covid-19 Host Genetics Initiative, which we contributed to very substantially from the UK, from both the Genomics study, which contributed many thousands of critically ill patients, and the UK Biobank study. An analysis that they've actually just reported last week used this Mendelian randomization trick to ask the question, does smoking cause worse COVID? And the trick they use is to assume that you were randomly assigned to your genes at conception, and that we already know that some genes make you more likely to smoke. So there are genes that govern your response to nicotine that make it more likely that you'll get addicted to cigarettes and smoke. And those genes that make you more likely to smoke altogether are associated with a slightly greater risk of getting COVID, which suggests that smoking itself is causing worse disease.
Isn't that a bit problematic though? Because just because you have a gene that may show an association with the likely that you will be more likely to smoke, that doesn't mean you will necessarily smoke.
Yes, you're absolutely right. The hope in studies of that kind is that those like factors like that one will wash out in the numbers, just because we have such vast numbers of patients, particularly in genetic studies of things like smoking, where there are literally hundreds and hundreds of thousands of people in the studies, that the fact that whether or not you smoke is obviously a composite outcome of many different factors, will just be overwhelmed by the numbers. And if the signal is real, then you'll detect it in the end if you have enough people in the study.
What I'm really intrigued about is how scientists account, when you're doing big studies like this and you have to be sure that you're accounting for all the other factors that could be affecting your results. How do you account for them in a society that is so complex, in which, for example, last year I remember studies that claim to adjust for socioeconomic status, and that can vary even within a family. You can have people of different socioeconomic statuses even within a family. So how do you do that, and how can you be sure that you're presenting something really value-free to the public?
So the trick is randomization, and the risk is that there are flaws in your randomization, that whatever you assume to be random is in fact not random, or that there's another pathway that actually explains why that random thing caused the outcome. So it might be that the genes that change your response to nicotine actually also change some completely a different part of your biology. Maybe, like Kirsten was saying, they change the response of your immune cells to the SARS coronavirus. So if that happens, then we'll draw the wrong conclusion, because we're only drawing conclusions about the biology that we know about. So the ways that we can correct for that are choosing randomly varying elements that are really random, like assignment to a given genetic variant, or like assignment to a given treatment arm in a study, and then watching carefully for other explanations. And it, in science, as Mike's already said, we make progress based on the best evidence, and we're often wrong. The process of scientific advance is learning where we've been wrong, and always recognizing that some of the theories that we're working from, some of the evidence we're working from, is going to be contradicted in the future.
I mean, certainly genes aren't assigned randomly, are they? They are clustered in certain in families and in populations. So, for example, if your dad smokes, you may be more likely to smoke, and you will share genes with your dad.
Well, yeah, so that's right. So the relatedness you can track, though. If you've done a genetic study, then you know better than anybody else in your study, but better than the patients themselves, who's related to whom. So that you can track and correct for. The fact that the environment in which you were brought up, the people who brought you up, influenced your outcomes, is of course not random, but you can correct for the fact that you're related to people who have the same genetics. So that in that sense, the randomness the assignment to genotype is still robust. The many factors in your environment, both the one that you mentioned, and lots of factors in your biology and in your environment that we can have no understanding of, are all taken account of by the fact that if you've got enough people in the study, you have equally matched groups of people who have either one variant or another. The part of the randomness that's not independent is nearby genetic variants, which tend to be inherited together. So, we haven't so far in this panel talked about the scale of the genome or how it works, but there are, the genome is made up of sugar bases in a line. There are only 4 bases in the code, the designated letters A, C, T, and G, and that's the code that makes your DNA. There are 3 billion letters in that code. And in little groups, the variants that are associated with one outcome are inherited together with other variants that lie nearby in the same little bit of genome. And where that happens, we often can't tell apart which of those variants are having a causal effect on your biology, and which are just passengers that are being inherited together.
Thank you.
Actually,
yeah.
Yes, while I think with Michael just talking there about the genome, one aspect that we haven't really talked about yet is that, of course, every single cell in the body has the same genome, but different cells use different genes to perform their function. So it's also very interesting to see, we've talked about all these risk genes that have been identified, but the next step is really to understand in which cells in the body do they actually function. And for COVID, that has really been very instructive, because when we first made the Human Cell Atlas, and then as early as April 2020, we were able to look at all the cell types in the body and say, well, which cell types actually express, for example, the viral receptor, and does that explain some of the pathology we see? And actually, it turned out that there was a very good correlation. So, of course, we know that the main pathology is the lung, and indeed, there's one particular cell type called an 82 cell, which has very high levels of viral receptor, and that takes up the virus and then dies and causes some of those problems. But we also saw some expression of this receptor, for example, in the enterocytes of the gut. And again, there are symptoms of diarrhea in many patients. Certain cell types in the heart also express this receptor. So again, we can use genomics here to understand not just the risks for getting disease, but also trying to understand some of the disease progression and the pathology that we see in the patients.
All right, so there could be genomic factors in different cells in different parts of your body that could explain why different people get very different symptoms sometimes.
Yes, I think that very different symptoms probably have to do with comorbidities and the very different responses of the immune system. But if we're ever trying to think about how are we going to treat the disease, or how can we stop the spread of the virus, to stop it getting into the cells in the first place, it obviously is really, really useful to know where the virus can go. So where we need to stop it from spreading, if you see what I mean.
Well, I'm going to go to the Q&A soon, but the last thing I wanted to ask was about long COVID, because this feels like the most mysterious part of this virus, and it's something that's affected so many different people in such completely different ways, and doctors don't seem to have a very good handle on that. So Mike, just going to you then, can we get any insight into long COVID at the moment, given what we know about the body and genomics?
I think there are sort of several competing hypotheses about the sort of the basis for long COVID and why it is that some people recover and some people don't. And I agree, it's really critically important, particularly when many of the individuals who are affected by this now chronic condition are people who were previously extremely fit and healthy. So it sort of goes against a lot of our prior beliefs about how comorbidities that are related to severity these things don't necessarily relate to the fact that individuals have sort of a chronic or an inability to properly or completely resolve the symptoms of their infection. Now, at the moment, genomics hasn't touched really on the long COVID question, and I think that's because at the moment it's very hard to define long COVID, and there are people, there are scientists, very well-respected scientists in the UK and around and elsewhere around the world who really are trying to answer that question of what is long COVID, trying to characterize it, trying to understand it, and people may have heard the name Dr. Nisreen Alwan, who's based at the University of Southampton. She really is a leading figure in this area, and also over in the States at Yale University, Akiko Iwasaki, who's an immunologist, is really trying to drive this forward. And in the UK, Professor Danny Altman, who's an immunologist. So at the moment, the focus is on sort of from the public health and from the immunology side of things. And one of the hypotheses that is gaining a little bit of support is that long COVID has similarities with autoimmunity or other autoimmune diseases. It affects the whole body. It's systemic. And there is evidence to suggest that patients with long COVID have antibodies that react to their own bodies. So it might suggest that this overwhelming infection triggers something in their immune system that was already there to recognize and attack their own bodies. But I think, this is very much sort of early doors in that respect. Genomics is very, very good at answering questions when you have maybe an idea about where to look, so which part of the body to look in. So with a respiratory infection, you need to look at the immune system and you need to look at the lungs, and as Kirsten suggested, also you need to look at the gut and look elsewhere. And actually efforts like the Human Cell Atlas, where we can start to get a sort of almost body-wide view of things, are extremely useful and be able to point us in the right direction. I wish we could say a bit more about long COVID, and it's something we're very much interested in, but it's sort of a bit too early to give anything concrete. And I know there are genetic efforts to try and unpick at least some of the molecular causes, or something that might give a hint as to what long COVID is, whether it's one condition or whether it's a sort of a cluster or a constellation of different conditions.
Great, thank you. Kenneth or Kirsten, do you have any thoughts on long COVID? Kirsten?
No, I have to say, I think the whole scientific community is still quite in the dark about long COVID. I mean, there are other viral diseases that also cause long-lasting fatigue that has been described for decades, and yet people still don't really have any answers for those diseases either. So, but I think with COVID clearly having these long-term effects, I think this is going to be an area of really active research going forward.
Okay, brilliant. Well, we've got some questions here, so we'll just get stuck into them. The first one is from Brian and Gwen. They asked, could COVID-19 have corrupted from a normal virus? That's implying that it's not a normal virus. So who would be able to take that one? Kirsten?
Well, I mean, it's quite difficult to describe what a normal virus versus an unusual virus is, but there's very strong evidence that the coronavirus that's currently circulating in the population is very, very similar to a virus that was identified in bats. And so the most likely hypothesis is that this virus from bats changed its host from bats to humans, and because humans have not had this virus before and are therefore not used to fighting it, the virus has caused this pandemic. And this changing species is not an unusual thing for viruses to do. We talked about mutations in the host, but obviously viruses mutate very rapidly as well. And if the molecules they use to latch onto cells, to latch onto certain receptors, change in subtle ways, that can mean that they now are suddenly able to infect humans instead of bats. This is a very well described process, and really not controversial that this can happen. So, for my money at least, that's the most likely explanation of where this virus has come from.
Okay, Michael or Ken, any thoughts on that question? We'll move on. Okay.
I think the scientific consensus is based on the evidence that's sort of in front of us, and the robust and rigorous evidence to that effect. I think supports exactly what Kirsten said. It's this zoonotic transmission from bats. Now, the exact ancestral virus necessarily might elude us, but the weight of evidence does indicate, as from my limited understanding, I'm not a virologist. My limited understanding is that is the case, yes.
Okay, we've got a great question here from Brian Connellan. Have there been any studies on the effect of COVID on individuals with identical genomes? So I'm assuming twins here, as would be the case with identical twins, he writes. And if there are differences, how significant might the differences be in the microbiomes? So these are two different things here, though related. Kenneth, what are your thoughts on that?
So I'm going to disappoint you to start with, because I don't know. We don't have evidence. I think probably in the UK, our genomic study is the likeliest to find those sorts of observations, and we are using a technology called whole genome sequencing, which is a sort of deeper way of reading the entire code of the genome, rather than the normal, cheaper, more efficient technology that that looks at, for genetic studies where you only look at the variation. So that means that if we find pairs of identical twins with different outcomes from the disease, we will be able to see not only what genetic variants they have that sort of vary across the population, but also genetic variants that might be unique to one twin or the other. There are now 13,700 critically ill people in the genomic study. It's the largest recruiting study ever in UK intensive care medicine. And I know of several pairs of identical twins who are in the study, and identical twins where one twin's in the study and the other wasn't sick enough to be entered in as a case. We are looking at their genomes, and we might see some of those signals probably in the next few months, but so far, to my knowledge at least, we haven't. I've been on holiday for the last week, so if the team had found something in the interim, then I wouldn't know about it.
If you did find some kind of correlation like that, would you be quite surprised by it, given all the other factors outside genetics that have an impact?
Well, Michael already mentioned the concept of somatic variation. So I mean, this is a different area of science really from what we've been talking about, but it is really interesting that your genome actually does change as your life progresses, and the genome of some cells in your body are different from other cells. That was actually my first ever big paper during my PhD, was showing that a class of ancient retroviruses that have become incorporated into our DNA jump from one part of your genome to another during the course of normal life. It's called retrotransposition. So your genome is mutable. It can change, and identical twins don't have, although they start out from the same cell, the genome changes early. So there are genetic differences between the twins, and of course, as Michael said, there are genetic differences between the cells of the same person. You were taking me to task earlier, Angela, for how could I be sure that a variable was random? The trouble with an observation that you could make, even if we find a pair of twins where one got COVID and was fine, one was critically ill, and there's one gene different between them, and it's a gene that we, maybe Kirsten's work has shown is expressed on cells that get infected with COVID. Even then, we wouldn't really know, because we've only got one observation, and so those kinds of discoveries would take larger populations or detailed study of the cells from a pair of people to show that there really was a functional consequence of the genetic variation.
And Mike, just coming to you. Oh, sorry, yeah, Kirsten, go ahead.
One thing I was just going to throw in there, we of course always say that the genetic information within every single cell of the body is identical, and that is pretty true, except for two particular cell types of the immune system, the B and the T cells, because they make receptors that are meant to target bugs and pathogens. And the way they generate a huge diversity of molecules that can recognize things they haven't seen before is actually by taking bits of the genome that is very nicely aligned in a particular region of chromosome and reshuffle them in a really random way. And that means that even two identical twins will have very different, what's called repertoires of B and T cells that can recognize different microbes and viruses. So, you know, in answer to the original question, you know, what if you have two identical people that respond very differently? The answer probably is that the immune system simply has a different set of molecules to attack that virus with.
Okay, that's a two brilliant answers there. Kirsten, I've got another question for you since you're an expert in respiratory illnesses. Nick Hindle asks, my wife has a suppressed immune system, and a couple of years ago, everyone else in the household was ill with flu, but she just had a slightly raised temperature and a bit of a headache. Is there a chance she might respond this way to COVID-19 if infected? Thanks.
Yes, and so one of the things about COVID actually is that a lot of the disease, especially the very severe disease, only happens once a lot of the virus has already been cleared from the body. So one of the things when studying infections is you've got to be it's a big dynamic process. So you first get the virus coming in, then there's an initial response that will hopefully suppress viral repression. The immune system then gears up to generate lots of these molecules that are very specific to that particular virus. They then trigger further inflammation, and then if all goes well, there's a third wave that suppresses the immune system again to return back to the status quo. But whenever we see very severe pathology in COVID-19, and what people often term this COVID storm, cytokine storm, it usually is a reaction that isn't actually wanted by the body, but the body for some reason isn't able to shut down this excessive immune response. And so the immune response, instead of healing you, can actually give you the disease. And so what may well have happened, and really I'm not a virologist either, and so I wouldn't be able to put my fingers on here, but when a virus enters and causes very severe disease, some of the disease may actually be the immune system in a way overreacting. And so if somebody's immune suppressed, and provided the virus doesn't get out of hand, they might have an easier time overcoming the infection and going back to normal.
That's great, thank you. I just want to get through as many questions as possible. Linda P asks, and this is more of a policy question, it may be a bit unfair, but I'd like to get your response to it because I think scientists have entered this space now, so you should have a view. Should the government be doing more to make people aware of the different symptoms in vaccinated people? Mike, what are your thoughts on that?
This isn't something I'm aware of, to be honest. I guess the issue might be that an individual who, if we assume that so the severity or the severity of the response to the infection is in part sort of baked in at the beginning, so sort of predisposed. Part of the effect of the vaccine is that it reduces the severity of those symptoms. So if you were predisposed to maybe having slightly lower oxygen saturation such that you might have been admitted to hospital, Kenny's the clinician here, so he can sort of correct any faux pas on my part. But you might be admitted and put under observation for maybe a day, maybe kept in overnight, but otherwise, you sort of your immune system just needed a little bit of a help to basically finish clearing the infection, and you're sort of well enough to go home. Maybe someone who's vaccinated in that position, it might be that their body clears the virus and resolves the infection a few days earlier. So it might be that the severity of the symptoms is not as bad. And this seems to be the case in so much that at a sort of a population level, there is not a breaking, but a slight disconnect now between the numbers of cases and hospitalizations, or at least there were a few weeks ago, and then the subsequent unfortunate mortality. So the vaccines, I wouldn't say that they've broken the link necessarily, but it does look like they sort of help to decouple things a little bit. Now, there's obviously all of the factors that we've talked about today: age, genetics, behavior, and as Linda P is asking about policy, there's also the restrictions in place. So the vaccines are probably helping, but then they're also maybe competing to some extent with other sort of behavioral factors that might mean that people's risk is sort of being modulated in one way and the vaccine's pushing it in the other. So, on average, the vaccines are probably helping in that respect. But policy is although scientists, I agree, should have an opinion, policy making is the domain of politicians. And we can put forward the evidence and so on and so forth, but it is the politicians who actually make those decisions for better or for worse.
Ken, did you have any thoughts on that?
Well, like everyone else in the country, I have lots of strong opinions on matters that I'm not qualified to comment on, and I think this is really one of them. I would reiterate Mike's point that vaccines and the infection that's happened already has not broken the link between COVID and critical illness, or infection in the population rather, and critical illness. It's just decreased the rate. So if enough people in the population get COVID, then a substantial proportion or a substantial number of them, a small proportion, but a substantial number, will end up having to come to the intensive care unit for treatment, which is a devastating event for them and their families.
There is a question here that I think is high in the minds of pretty much everyone at the moment, from Suman Rai. She asks, is there a likelihood of another variant developing that could cause increased infections if it occurs and continues to increase? So we have, all these different Greek letters of variants, but how long will that go on and how many more letters will there be? Kirsten?
I think the answer is yes, that is clearly a possibility because we've already seen it happen. So, there was the first variant and then the beta variant or the Kent variant came along that was more infectious. And then, with a very huge viral expansion within India, a new variant arose, which was the Delta variant that is even more infectious. And there's no reason why the same thing could happen again. And that's why I'm personally a little bit worried about policies that allow a relatively wide viral spread in the population. But having said that, it will be within the UK, if there now are a lot more cases, I think that is somewhat worrying, but at least many of those people will have had a vaccine so that their ability to fight the virus is relatively better than of unvaccinated people. But for example, there is evidence that people who are immunosuppressed and where a virus might continue to replicate for a long time is actually able to sort of incubate the generation of new variants. And of course, people don't really, often you wouldn't necessarily know whether or not a particular person has this ability to fight the virus completely or not. So we wouldn't really know who in the population might be doing this. It's just a random thing. So we've been very careful not to express too many opinions on things that we don't really have a great expertise on, but having a very large number of people carrying the virus does worry me.
And I think, many of us feel now that we're having the vaccine, we'll be safe, that we're far less likely to suffer critical illness or pass away. But now that we have a vaccine, will it work against pretty much any variant that comes along, or will it continue, or would it be possible for a variant to come along that would be vaccine resistant, for example?
So I guess this is in part trying to predict the evolution of the virus, and evolution is by itself, or the fuel for evolution is mutation, which is in itself a random process. So it's potentially very dangerous to try and predict the sort of the specificity or specific nature of something like, this variant will arise at this point, or it might arise in these particular conditions. Now, the other sort of material components of evolution, or the way anything evolves, any genome, be it a virus, an elephant, a mouse, literally anything, it could be a banana, the sort of the evolution of that lineage anyway. The point is that you have mutation as the fuel, but the thing that drives it or shapes it is the natural selection itself. So the situation that Kirsten described, where you might have an immunocompromised individual, that sort of creates a setting where there's no longer a selection pressure to remove the virus. It can kind of basically freely mutate and accumulate things. And then a particular viral particle that has a very specific genome might find itself in a situation where its genome gives it a slight advantage, a growth advantage, for instance. Maybe it replicates more easily, or it more easily infects the host cells. And this is probably how the variants that we have arisen. We didn't predict that any of these would arise in this very specific way, but they can. And I think that's probably the possibility is the thing that's actually really important, is that we know that possibility exists, and there are mitigations that could be put in place to help alleviate it, or to help reduce that risk. So if I were to put forward an opinion, my personal opinion is that we should be putting in place things to help reduce the probability or the risk of those variants arising. Now, we don't know the impact of any given variant, but I think the more variants there are, the higher the probability one of them could be sort of worse off than the Delta variant, for instance.
But what kind of measures? Sorry, Kirsten,
one thing to add there maybe briefly is, though, that so obviously a lot of the vaccines are based on using the whole of the spike protein, and that on the surface can mutate and can change. But a lot of the internal structure can't really change that much, because the protein needs to stay functional, and that puts certain constraint on it. And when this whole protein is seen by the immune system, even a variant that has some new changes will probably, as part of that protein, also have areas that stay the same, that do not change. And therefore, there's also a good probability that once you're vaccinated, you can respond even to new variants. Maybe not as effectively, but a lot of the viruses a lot of the vaccines that were based on the initial isolates have still protected against the Beta and the Delta variant. Maybe not quite as effectively, but so we don't need to worry that. Or I think there's also a good chance that the vaccines will give some level of protections even when new additional variants arise.
Okay, that's very reassuring. That's kind of what I was looking for, really. And Mike, then just going back to you mentioned measures there in order to protect against the risk of these mutations occurring. What kind of measures would you like to see?
So one of the things that you can probably do is to get vaccinated. It's one of the things that we've talked about the fact that it helps to protect against the severity of the disease, but also to some degree, and I don't know what the exact number of this is, that it can reduce the probability of being infected or infecting someone else as well. So the vaccine is the simplest way to help mitigate that, or mitigate against the infection and the severity. Now, the rate at which the population becomes vaccinated is also important. So from an evolutionary standpoint, the virus is mutating, and I talked about selection pressures. So one thing you can do is, if you vaccinate very quickly, you create essentially such a sufficient selection pressure that you can sort of massively reduce the chance of particular or sort of from the variant. You don't give the virus time to kind of evolve and accumulate mutations, essentially. So, you know, the rapid rollout of vaccines, which to some extent has happened in this country, but now looks to be tapering off. So, you know, I really would sort of definitely encourage anyone to get vaccine. Now, people will have, some people can't be vaccinated for various reasons, and there is a lot of hesitance, or there may be some hesitancy in terms of, you know, do I want this vaccine? What does it mean? What is it going to do to my body? And those are entirely valid questions. And so the medical community is there to kind of answer those questions. So if you do have any questions or about the vaccine, then those are things that you can sort of ask a doctor, or you can go to the vaccine center, you can talk to them about as well. So I really do think that is one of the best things that we can do. The other is maybe a little bit sort of maybe blue skies, is that we can do things to stop zoonotic transmission of viruses as well. You know, there are sort of very high level studies suggesting that the encroachment of humans on habitats is going to increase the risk of these zoonotic transmissions in the future. So understanding how our behavior impacts on biodiversity and how it impacts, and that subsequently feeds through to the interactions between humans and the rest of the animal kingdom, what that means for our public health is really, really important. And again, I'm not an expert in this at all, but it is an area that is perhaps underappreciated in the public domain.
Okay, that's great. And just for the people in the audience, if you could please put your questions in the Q&A, because I'm not reading the chat, I'm afraid. I'm just looking through the Q&A questions. So if you have one, then put it in there. We have a couple of questions here, which I'm going to read out, even though the questioners themselves have said they're stupid questions. But like I said, there are no stupid questions here. One of them is it fair to think that the better the immune system, the worse the resulting variant is likely to be? So I assume they mean your response to the virus is likely to be. Like if I had been strong enough to fight, if it had been strong enough to fight its way past the body's heightened defenses, does that mean it itself has become stronger and altered enough to be a further problem? So I'm assuming this questioner means what, you know, if your body is able to mount a very robust defense to a virus, if it does manage to get through, does that mean you will suffer worse symptoms as a result? Kenneth, do you have any thoughts on that?
Yes, so I'm not an evolutionary virologist, so I'll present that disclaimer before I comment. But if you imagine a strong immune response, you know, as is one in which you clear the virus very quickly and don't get sick, and if there's not a lot of virus around, then the virus doesn't have time to change. The situation that Kirsten described, where the virus persists in one person for a long time, is the one that everyone worries about for the evolution of new variants. Because if the virus is making copies of itself, you know, many times those copies have a mistake or a mutation, and eventually, just by random chance, you know, sometimes a new variant emerges by that process. So the risky situation is when there's lots of virus in the population, and you know, in an individual, just copying itself, because that's what enables the virus to change. And again, with the disclaimer that I'm not an evolutionary biologist, now I would be interested to hear what Kirsten thinks about this too. I think it's important to recognize it's not trying to kill you. It causes severe disease. The disease that we see in an intensive care unit is primarily an inflammatory injury to the lungs. It's made the body's own immune system that's causing the damage. And the virus, that there's not really any imaginable way that could help the virus. Doesn't really help it to transmit itself like a cough or a sneeze does. It's just causing the patient to become desperately sick. So when it mutates, the mutations that will come under positive selection in the viral population, there's no reason why they would be mutations that would make you more sick, or at least no reason that I'm aware of. But of course, it might well be that they evade the vaccine or increase the virus's ability to copy itself. And there's a possibility they might cause worse disease, but it's not what the virus is trying to do. It's not what evolution is selecting for.
Okay, brilliant. Kirsten, what are your thoughts?
Yes, I very much agree with everything that Kenneth has just said. And again, I'm also not an evolutionary biologist, but I think it kind of makes sense that if there are huge pools of viruses out there that are all just mutating a little bit, but just by the sheer force of numbers, you know, if you have a lot of virus, you are more likely to somewhere along the line get a variant that somehow turns out to be more harmful. And I think it was Anthony Fauci who put it very nicely and said, we aren't safe till we are all safe. And what he was really trying to refer to is that vaccination in one country really isn't good enough. It's got to be vaccination around the whole world, because if we vaccinate everybody in England, but we allow we stand by while other countries have huge third and fourth waves of the virus, new variants are more likely to arise, and that clearly isn't good for any of us.
Wonderful. We have about 10 minutes left, and I've got a question here from Mike, I think. This is from Jorge Aponte Gomez. He asks, is it possible to have a computational model of the immune system to address viral infections and their results? I imagine that's your dream goal one day, that you could just map the immune system in some way using computer models. Are you anywhere close to that?
I mean, we sort of variously model different aspects of it, often to answer very specific questions. And the huge efforts that the Human Cell Atlas that Kirsten's sort of been one of the driving forces in really is a way for us to gather the necessary data to be able to move towards that goal of modeling a cell, to modeling the interactions between cells and what have you. One thing I would say is that there's a quote that is attributed to a particular statistician. I forget who it was exactly, but all models are wrong, but some models are useful. So generally what we tend to use them for is to be able to look to see how something's behaving, compare it to our observations, and then be able to develop new experiments and develop new hypotheses about how something in particular is working. And now, whether we could have a model of exactly how all of the nuts and bolts of the immune system are working together in every single context, it's sort of a very grand idea, and it's maybe somewhat akin to sort of a unified theory in physics, although it's a bit more concrete in physics, because not that the maths is easy, but the sort of the situation you're dealing with atoms and subatomic particles and that sort of thing. Whereas with cells, you're dealing with things that are made up of all of those things. It's thousands, orders of magnitude larger and more complex. So it would be fantastic to have a working model of the immune system, but then that would probably put us all out of a job. So maybe not so good.
And is it also complicated by the fact that we as individuals are so different from one another that even if you can model one person, say, that doesn't necessarily tell you much about the next one?
It is a huge problem, and it's something that we sort of battle with to some extent. And some of the work that I'm very much interested in is being able to how can we actually sort of use that variability between individuals, between cells? How can we use that to really understand how things are working? And some of that variation is genetic, and the work that Kenny's done, and some of it's the environmental things that we've talked about. Some of it's differences between children and adults, as Kirsten suggested as well. the one thing that's particularly maybe important and to sort of take home message is that we should never underestimate the importance of stochasticity, so random factors. There is always a degree of variability in anything, and particularly so in biology. And so being able to predict something is only ever going to be able to predict it with some degree of uncertainty attached to it, basically. It's, yeah, very hand-wavy, but that's kind of maybe a statistician's answer.
No, that makes sense. Kirsten, just coming to you then, because you deal with respiratory illnesses more broadly. How does that, how do you deal with the fact that people are so individually different when you're trying to get a grip on a new disease?
I think to a large extent, we just have to hope and pray that the immune system we're endowed with is able to cope with the unknown, and the whole the way the immune system has evolved is exactly so that a new disease that emerges can still be fought off. And the best, I guess, we can do is sort of support the immune system. And as we are getting better at understanding how to sort of either put the accelerator on and rev the immune system up a bit more, or for example, at the end of the response, like we've just said, when there's a sort of pathological immune response, if we are better able to understand how we can rein in that sort of excessive immune response, I think that's probably, those are the most promising ways of dealing with new emergent infections that we haven't seen before.
And another question for you, Kirsten, I want to go to first. Why does the vaccine not work for some people? I know this is quite rare, but why is that?
I'm not sure that it's really completely understood. Again, as I outlined earlier, we're not born, the immune system, especially the specific immune system, the antibodies that people form, they aren't the same in everybody, and everybody makes their own set of different antibodies, and there's a second molecule called the T-cell receptor, which is similar to an antibody. In other words, a molecule that has a huge diversity within the body and can recognize all these different bugs and viruses. And because of the randomness of how these molecules are formed, it might just turn out that within the repertoire of antibodies a single person has, there just isn't one with a particularly good fit. So you then just don't mount a terribly strong response after you've been vaccinated. And therefore, when the real virus hits, you're still not terribly well protected. But there are many other factors that can play into it. As we've said before, the immune system is very complicated. And so I wouldn't want to speculate on this for too long.
Sure. I don't know if anyone can answer this, but if, let's say, there were a patient for whom one vaccine didn't work, are the different vaccines different enough that another vaccine would work on them? Kenneth, do you have any thoughts on that?
So, in vaccinology, I think, as Kirsten explained, the reasons why a vaccine response might not work in a person, given with our model of the disease, might be because they don't produce antibodies. It might be because they're particularly susceptible to just an overwhelming amount of virus that replicates in their system, and that overcomes the immune response that's there. So it on the reason why the first vaccine hasn't worked. The vaccines that are available operate through a variety of different mechanisms. If the reason for failure related to the mechanism of action of the vaccine, then different categories of vaccine would work.
Okay, great. There are a few questions here about long COVID. So I'm just going to read out one of them, but they're kind of grouped together. Essentially, people are trying to get a grip on how long COVID manifests and what it really means and why it's so different for different people. But Suman Rai asks, I heard that scientists are tending to group symptoms into two areas when it comes to long COVID, respiratory or nose and throat, both with or without fatigue. Does anyone have any comments on that? Any thoughts? Kirsten?
I'm really not an expert on long COVID, so I would prefer not to comment on this one.
No, that's fine. I mean, what does intrigue me, we've got a few minutes left, and I think a lot of people are kind of scratching their heads over what is long COVID, how does it manifest? Is it just the case that this is one of those conditions that doctors really have no idea about, and so they've just given a label to this set of symptoms, and only the future will really be able to give us any kind of insight into whether this is a specific condition or something else, or as Mike, you said earlier, a cluster of conditions?
I think perhaps it's just on the record to note that the name long COVID came from the patient community itself, and there has been a huge grassroots movement that it has emerged over the last year, because all of these individuals were infected with the coronavirus and didn't recover, and they found each other through internet support groups. And that from those groups, the name long COVID was born, and they've really have been helping to push some of the research efforts in terms of characterizing what long COVID looks like. I don't know the necessary details. So it's come from the patient community rather than from the medical community. And I think perhaps we're in somewhat uncharted territory here, in that maybe people from other patient communities might disagree with this, but it's unusual in that the disease is being characterized initially by the patients and not by doctors and by clinicians. Now, I personally think it's very important for the two communities to work together. The patients have got the passion to understand the disease as much as the clinicians do, and the clinicians have the resources. They have access to research funding. They can talk to scientists. They can build the studies that are required to be able to draw robust and rigorous conclusions in this area. Some of those scientists, some of those clinicians are sufferers of long COVID as well. They're part of that community. I generally think it was so early on in the process of understanding what it is, and I'm sure that is extremely frustrating for people who have this unknown condition that is sort of affecting them. So although it's not a very satisfying answer, it's probably the most accurate that I'd like to give at the moment. I know, Kenny, we've sort of had various discussions about long COVID in the past. I don't know maybe how things are sort of evolving in the Somatic community or in genomic with that respect. Is there anything you can comment on?
So we've no new result, and I share your inspiration from the long COVID community, who have the patient community who describe the illness. I think we the last year and a half we've been witnessing the emergence of new diseases, and the scientific response to them, first to work out what the disease is, which in the case of SARS-CoV-2 was done by clinicians and scientists in Wuhan. The pattern of disease, and particularly at the critically ill end of the spectrum, is remarkably similar compared to other diseases we see in critical illness. So it was a relatively easy to identify and study. Of course, because of the 21st century science, we were able to positively identify the bug that was causing disease incredibly quickly, and by the time cases started arriving in the UK, the virology lab telling us which patients had COVID and which ones didn't. So it was a acute COVID, which the opposite of long COVID. It was a relatively well-defined group of patients. So studying them with genetics is easier than other diseases like sepsis and influenza in critical care, where there's much more variation in the patient population. Long COVID,
maybe I could make one comment.
Yeah, please.
Yes, go ahead.
So much as I wouldn't want to speculate what causes it, but maybe on a slightly upbeat note, I think the speed with which the scientific community has really found out what SARS-CoV-2 is, how the body responds, really gives me great hope that we will also understand long COVID really relatively quickly. You know, there are a lot of the techniques that we've applied to the sort of acute COVID that we can also apply to patients suffering from long COVID. We can look at all the different cell types, how they're activated, whether they're replicating more quickly than they should. And we can look at the antibody repertoires now in a lot more detail than we could previously. And I would also say the funding is available to carry out relatively large studies so that we can deal with some of the heterogeneity between people that we were talking about earlier. So much as we don't know that much as yet, I think the huge progress we've seen over the last 18 months really gives me hope that we will find out what causes this, and hopefully with that we will also start to be able to come up with some interventions.
Oh, well, that's a very positive note to end on. I've been really cheered in this event that we've had scientists who have been happy to admit what they don't know as much as what they do. And I think for me, at least, it's been really enlightening. I've learned so much and I feel quite reassured that my vaccine will keep me safe, which is good. Well, thank you everyone for listening. I'm going to hand back to Kenneth Skeldon now from COVID Connections. But thank you to our wonderful speakers.
Thank you.
Thank you very much, Angela, and I hope everyone found that an interesting discussion. I mean, I think it's really interesting how genomics, the science of genomics, is not helping just with the kind of surveillance and the tracking of the virus, but also in thinking about these quite complex issues around how it causes disease and the kind of symptoms that people experience. We talked a lot there about the genome of the virus and its genetic code, and I just thought it was interesting to highlight that Andrew Steele, who's a British science writer, has been doing some work on mapping the genome, and find out that if you reduce the individual letters of the genetic code to about 2-point font, you can fit the entire genome of the virus onto a single page of A4 paper. And then the very, very small variations that happen, the mutations, when they happen in key places, produce these key variants. And this is the Delta variant here. So if people want to sort of investigate that and just find out for themselves a little bit more about the kind of research behind the scenes that's going on, that's a really nice resource as well. As is always the case with these events, there are so many questions, more than we could get through in the time. But hopefully we managed to cover lots of the themes that were coming through in the chat. If you'd like to be in the audience for our next COVID Connections talk, that will happen on Wednesday, the 11th of August. When we'll be exploring how important that the sharing of data has been in advancing our understanding of the virus, and how important it is for people to agree to share that data. So the kind of role of the public in that. Also, some of you might be interested in the event that's happening next week, which is part of our Genome Late series. That's on Thursday, the 22nd of July. The theme for that event is aging, and in particular, whether genetic insights from the animal world can shed light on how and why organisms, particularly mammals, age at the rate that they do. We'll put links in the chat. We're doing that just now with the website to all our events, so that you can sign up to receive our newsletter as well, to keep you posted of things that are coming up. We'll also put links in the to the websites for where each of our panelists have come from tonight, and the COG UK website, so that you can read a little bit more about the work that's happening across the consortium. We've also put a link to Angela's website where you can see our latest books and work. If you'd like to watch this event again and share it with your friends, then we'll be posting that on our website in a few days' time as well. We'll also be posting a short survey. Please, it is important that we find out what you think about these events, because we would like to hear your thoughts. We're always looking to improve things. So if you can, please stay online at the end and just for a couple of minutes, fill out our feedback form. We'd really appreciate it. So finally, thanks to all of you for coming, and thank you to Angela, to Kirsten, Michael and Kenneth for joining us this evening. Take care, stay safe, and see you again soon.